Hello, everyone, and welcome to October's Ask the Expert session. I am Mark Chatterley. I work at PSC Support. Usually it would be Paula sat here, our Chief Executive. Unfortunately, she can't make it tonight, so I've stepped in. Today, we have got the wonderful Professor Iqbal from the Queen Elizabeth Hospital in Birmingham. He's I hope I say this right, a professor of clinical microbiome. See, I did stumble over that. He is a wonderful clinician. He doesn't do clinics anymore, which is a shame for me because he was my gastro doctor, very useful. So I'm a little bit biased in this, I have to say, but I think you're wonderful. You're
Okay.
going to you're going
I
to talk
thought
to
you
us.
guys, sorry.
Well, yeah, you got to have a little bit of praise to start these things off. And he's going to talk to us today about PSC-IBD, PSC-IBD, which is, I believe, the best way to explain it is it's kind of the new name for the type of inflammatory bowel disease that PSC people tend to have. I'm guessing that's not always the case, though. But I will pass over to you. I believe we've got a bit of a presentation, and then we can have some questions at the end if you want to fill in the Q&A little taskbar.
Yeah, thank you very much, Mark, and thanks very much for asking me to do this. I have to say that I've been doing the PSC-IBD clinic at the Queen Elizabeth Hospital for the best part of a decade, really.
Yeah.
And I've now retired into research, but I've got only because a very good fellow has now finished his PhD, and he did quite a bit of
Excellent.
work on that, so he started. But I have to say that it's the most stressful clinic I've ever done, really.
You have to deal with people like me. That's why.
No, no, not at all. The people, the patients are wonderful, and I really enjoyed that aspect. But it's just because it's so important managing the
Yeah.
IBD as PSC, because I don't really think of PSC and IBD being different. I believe
Okay.
you're intimately linked, and the IBD actually, to a large extent, in a lot of them, this condition is driving the liver problem. So, and you mentioned microbiome is my main thing, and so I'm going to not talk too much about the immunology of PSC, but I
Okay.
need to focus on the microbiome aspects really. Um, so yeah, what is PSC-IBD? I kind of, can you see these slides? Yeah.
We can. Yeah, they're on my screen now, so which means everyone should see them.
Yeah.
Yeah.
Okay. Okay. So yeah. Well, let's start with the gut microbiome, and this is obviously become something of great interest in the last 20 years since the advent of genetic ways of investigating this, because intrinsically, if you take a stool sample, pretty much all of it, all the bacteria in there will die as soon as you produce it, because they don't live in oxygen. And in fact, this is a this is a may as well go large here. This is the beginning of the Earth 4 billion years ago, and this is where we are now. And until 2.5 billion years ago, the oceans were full of single-celled organisms, bacteria, fungi, the microbiota, and then 2.5 billion years ago, the vast majority of these early life forms died off as a result of the production of oxygen by early plants and also oxygen producing microbiota in the oceans. And so the first multicellular life forms which have been discovered from the fossil records from about 600 million years ago. And as you can see, this Girona piana is actually one of the earliest in the fossil record of a multicellular organism, and it's pretty much a gut. And there are two reasons really for the development of multicellular organisms, which then led to animals and the rest of the animal kingdom. One was, if you have a load of cells put together, you're less likely to be eaten. And the second is to provide a home for the microbiota, the bacteria, fungi, and other components of the microbiota, which don't survive in oxygen. So this gut, this anaerobic environment for the as a home for bugs is a really ancient, really very ancient thing. And so as a consequence, the relationship between those I'm going to call them bugs because it makes it just easy. It's not
I,
scientifically correct, but the bugs which live in our bowel and our bodies as we've evolved and our immune systems is a really ancient and complex relationship which we're only just beginning to understand. And so just some terms that the microbiome, the microbiota are the microbial organisms which make up the microbiome. So they're not just bacteria, but also archaea, viruses, and fungi. And we know pretty much nothing about most of that, apart from the bacteria, really. The microbiome is an encompassing term for the whole community of microorganisms, including their genetic material. So that implies a functionality. It allows you to understand, because we were looking at their genes now, the microbiome is the whole community and what the community can do. And this is a horrible word, which microbiologists and ecologists really hate, but it's become part of the vocabulary. So you'll hear it a lot more. The dysbiosis is a state where the normal microbiome is, first of all, reduced in terms of diversity, and secondly, imbalanced in terms of makeup. So why have a microbiota in your gut and a microbiome? Well, it's very much a symbiotic relationship. So in the context of the colon in particular, nutrient metabolism is really important. So there's a whole bunch of microorganisms which synthesize secondary bile acids from primary bile acids. And this is likely to be very important in primary sclerosing cholangitis and the liver side of this. Although we don't understand at all how it works, this bile acid synthesis and the action of bile acids is going to be very important. And then the anaerobic or oxygen-hating bacteria in the colon are very important for the digestion of the kind of fibers which we can't digest with our human enzymes. And the digestion of these fibers in the diet provide short-chain fatty acids, which are really important for the health of intestinal cells and have anti-inflammatory properties as well. So that's really important. And one of the problems with the Western diet is we don't have enough of these fibers. As a consequence, our microbiome, anaerobic microbiome, is degraded. And then if you don't have provide the food, the microbiota start eating the mucus layer in the gut. So that's one of the issues really in IBD. Then obviously the relationship between the microbiota, the bacteria, and the host immune system, or the immune system of our gut, and by extension of the rest of our body, is really important. And there's a careful balance which is out of kilter in chronic or long-term inflammatory conditions. And the microbiota are also involved in metabolizing chemicals and drugs as well. And certainly one of the drugs which has been used for inflammatory bowel disease for many years, 5-aminosalicylic acid, nazalazine, does depend on in certain formulations on the action of microbiota to digest the drug and make it available. And if you have animals which don't have a microbiome, so germ-free mice, they have huge problems with the development of their immune system. But also the other side of it is they don't actually develop inflammatory bowel disease unless you put bacteria into their guts. So diversity is really important. And the alpha diversity is a measure of the diversity between two populations. If you look at the microbiome, I'm not going to labor this, but essentially diversity in anything, government, politics, football, is a healthy thing. And once you lose diversity, then things start to go wrong. This is just a sort of simply this is just to explain how we look at bacteria in the gut. So this is a human taxonomy. Every organism is classified according to which kingdom they belong to, which phylum they're in, what class, what order, what family, what genus, and what species. So we belong to the family of animals. We are the phylum Chordata. We have a backbone. We're mammals and do all the things mammals do, like give birth to children and breastfeed and so forth. We're primates, hominids, Homo, And the only species remaining on the Earth is sapiens, although I'm sure we've all seen a few Neanderthals occasionally, but then they're not really Neanderthals. Homo species, Homo sapiens is the only species. But importantly, most of the literature in this space, when you look at the papers around it, use a genetic technique which is cheaper looking at this form of, which allows you to separate humans down to family level pretty much. So you can differentiate between humans and gorillas, but it's not until you get down to metagenomic sequencing, which is becoming more common now, where you can look at the whole genetic structure in a system where you can actually start differentiating down to species level. In the gut, we have 65% or a majority of firmicutes, which are they're called gram-negative because that's the way they stain when you stain them under a microscope. But these are anaerobic organisms which don't survive in oxygen. And they're important for digesting dietary fiber to make short-chain fatty acids. Bacteroidetes are the other main, again, anaerobic. They don't like oxygen, and they produce, they help in nutrient digestion generally. And then it's only 5% of the microbiota are made up by other phyla, such as the aerobic or microaerophilic, in other words, bacteria which can survive in small amounts of oxygen, and they're generally the bad guys. The salmonellas, the E. coli, the Shigella, the pathogenic, the stuff that gives you diarrhea, which stimulates your immune system.
So,
Again,
just to clarify, just so I might have lost myself a little bit there. So if you take a sample from any human's gut at the phylum level, they differentiate into those three groups.
Those three groups largely, yeah. And the majority of them are anaerobic or oxygen, bacteria which can't survive in oxygen. But the ones that like oxygen are the ones that damage the gut. And those are the ones which get overexpressed or they're overpresent in inflammatory bowel disease. And just as a sort of point of reference here, just to show you how early we are in our understanding of all this, if you're looking at a phylum level at a human being, then you can just about distinguish us from sea squirts, reptiles, but no more than that. So again, it's quite a high level data. This diversity really matters. So the Baka pygmies are the original, the only sort of rainforest hunter-gatherers now, and they have a very nice, diverse, rich microbiome. If you go to a local agricultural community like the Bantu, immediately the diversity of the gut and the richness is reduced. And if you look at Americans, in particular Western Americans on a high-fat, low-fiber diet, the diversity is greatly reduced and much more prone to chronic inflammatory diseases. And so most chronic diseases, including gastrointestinal disease, are associated with loss of alpha diversity. And if you look across species, so within the primates now, humans have the worst diversity in the gut, and of the humans, then the Western gut, the American gut, is particularly poor in terms of diversity. And this
would that be, sorry, I'm jumping in again. Would that be because we're not exposed to as much of those germs and therefore they don't take hold, or is it a factor of our diet not requiring them to be there?
Yeah, there's a factor of our diet not requiring them to be there.
Okay.
And a lot of studies of people who migrate from one area to another, it's an environmental thing really. So there are many environment factors which affect the microbiome, and diet is by far the most important because that's pretty much maintained through life. So yeah, it's a dietary thing. Thank you for asking. But this reduction in diversity and this dysbiosis, therefore, is a feature now which has been recognized in many chronic inflammatory conditions. You can see from IBD to chronic liver disease, chronic kidney disease, certain brain disorders. You can read them all there. This is an important pathogenic factor. And there's this American gastroenterologist, Dr. Blazer, has wrote this very nice book called Missing Microbes, which explains how these, certainly in mice model way, how important a diverse microbiome is in maintaining health. And there's this clear association in the 20th century between a reduction in infectious diseases, mirrored by an increase in chronic inflammatory conditions, with the advent of penicillin in 1920, in the 1920s. So in inflammatory bowel disease, which is the paradigm for this, really, here on the left here, you see a healthy gut with tightly bound cells, a nice thick mucus layer, a diverse, although it's not really diverse here in this picture, but a diverse microbiota living within the gut, and an immune system which is basically tolerant. All the immune cells here are mainly T regulatory cells, which are there they're anti-inflammatory, and they kind of signal that everything's happy. Once you get inflammatory bowel disease, then the mucus layer gets degraded, the cells become leaky, there is a translocation of these bacteria across the cell wall. The bacterial population is marked by a reduction in diversity, a reduction in short-chain fatty acid-producing bugs, an increase in these proteobacteria, these oxygen-liking bugs like Shigella, E. coli, Salmonellas, and instability. And as a consequence of that, the immune system gets triggered off. You lose your T regulatory cells. You get these they're just, I'm sorry about the labels, but these cells actually, they're inflammatory cells. They cause inflammation in the gut, and they produce all these chemicals against which we now have therapeutic targets. So TNF alpha, IL-20, 12 and 22, which are targeted by drugs, TNF, IL-23, and these inflammatory cells can also target other organs as well. And the paradigm is now that off-target effects, such as joint problems, liver problems, are probably actually programmed from the gut in terms of these T cells, and hence the intimate relationship between IBD and PSC. And now this isn't gospel, but there's a hell of a lot of evidence that just suggests this, and this is what I believe. So the treatment for IBD in PSC is really no different from the treatment of IBD in IBD without a liver manifestation. So basically, we tend to start with 5-aminosalicyl-mesalazine, then steroids as a short treatment to kind of bring the condition under control, immunomodulators such as azathioprine, 6-mercaptopurine, sometimes methotrexate, which generally reduce the effectiveness of these T cells, and then biological agents, which are targeted now as monoclonal antibodies against TNF alpha. So we have infliximab and adalimumab, ustekinumab, which targets IL-23 and IL-12 as well, and then the son of ustekinumab, risankizumab, other monoclonal antibodies, which are antibodies directed against these chemicals, are the mainstay of the treatment of inflammatory bowel disease. And they're very powerful, they work very well, but not for everybody, and the effect does wear off. And then there are some small molecule tablets which have come out now, which have a very powerful effect on all of this by acting on the cell. Things like tofacitinib, and you will have heard of these drugs. But essentially, the treatment algorithm is this. It's basically a top-down. But my message really for PSC-IBD is maybe top-down it would be better than bottom-up. So maybe start earlier, because it's really important the IBD is kept under control. So inflammatory bowel disease is driven then by whether it's PSC-IBD or IBD, it's the same thing. You have a genetic predisposition, which is there have been more than 200 genes associated with. There isn't one gene in most people. This is just the kind of general makeup. There is a disturbance in the microbiome driven by environmental factors, which are poorly defined, but clearly diet is very important. Smoking is another. And this leads to a degradation of the intestinal barrier and an immune response. And that immune response can either be just directed to the gut or the joints or the liver, and PSC is part of that liver paradigm. So ulcerative colitis is without liver involvement, generally affects the left side of the bowel. We all know it causes bloody diarrhea and is a hell of a problem. But it is rising in an epidemic way in the world. So if there are three things which are important, the immune system, our genes, and the environment, the immune system and our genes haven't changed in the last 40 years, but the environment has. So, and this is where change in the microbiome, I think, becomes really quite important. In chronic liver disease, PBC, PSC, autoimmune hepatitis, there is a similar reduction in diversity in the microbiome, an increase in those E. coli, enterococci, which are talking about, which are the those oxygen-liking damaging bacteria, and a reduction in immune tolerance as a consequence. The difference in PSC-associated IBD, and I'll show you some evidence for that in a minute, the bile acid synthetic pathways, the way you metabolize bile does seem to be more important in people who have the liver side of the problem compared to just IBD without liver manifestation. So, at least 80% of people with primary sclerosing cholangitis will have inflammatory bowel disease. Sometimes it's there from the beginning. Sometimes it comes after the liver transplant, and which we don't really understand why that is. And you can see here that it can either be right at the beginning of the disease or really even up to 5-10 years. Sorry, 5 to 15-20 years after the disease. The other side of it is that if you look at a cohort of people with inflammatory bowel disease and actually have a careful look at the liver by imaging, you will find 1 in 5 of those patients, people without any enzyme problems in their liver, will have some changes suggesting primary sclerosing cholangitis. And this is I don't want to put scary stuff up, but I think I just want to use this slide to just illustrate the point. If you have a liver transplant and you have inactive IBD, you're much more likely to hang on to the liver than if your IBD is active. So it's a really important point in terms of maintaining your IBD under control. It's very important, this. And you can see here from this Australian study that if you look at people who've had liver transplants, then if your IBD is mild compared to if it's severe, again, there's a big difference in liver health here. So, when I first started doing this clinic, my predecessor really didn't take the IBD very seriously, but for me, it's been really important, the IBD side of it, and Palak is always knocking on my door and saying, fix the IBD. So there isn't actually an active liver treatment yet, right, which is recognized for the things are in development.
Do you know how they are measuring the mild, moderate, and severe? I don't have a kind of a scope for how my IBD would be categorized, if that makes sense. Because to me, it's just horrible, or I can cope with it.
Yeah. So the I think the important thing, Mark, is that you need to look with an endoscope every year.
Okay.
Honestly, and it's not just for a cancer surveillance. It's there to make sure that it's okay and looks okay, because we don't have very many non-invasive markers of IBD. We have fecal calprotectin, and we have with CRP and stuff like that. But the thing is that most people with the form of PSC IBD, the disease tends to affect this side of the bowel, the right side, as opposed to the left side, and so the rectal bleeding and the sort of tenesmus, which is the sensation that you really need to keep going to the toilet, is not so much of an issue. It could just be a general feeling of not quite right, So I think if we have fecal calprotectin, we have to clinical scoring scales, such as Harvey-Bradshaw index for the more sort of Crohn's type of phenotype, where small bowel is also involved, Mayo scoring for ulcerative colitis. But I think that the key thing is keep an active eye on it, like do an endoscope every year or two, I think
So
it's really.
that sort of definition of mild, moderate, severe isn't necessarily something for us to worry about as long as we let you know what symptoms we're experiencing
That's it.
and
Yeah.
let the colonoscopy and all of that inform your decision making.
Yeah, and we use non-invasive markers such as fecal calprotectin and CRP and so forth. But I think it's really important to keep an eye on it, really,
Yeah.
so much. Yeah. So Nabil, who did a PhD with me and has run off to Dubai because of family issues, and it's a real terrible thing, was able to show that in PSC, patients with PSC and IBD compared to IBD alone, compared to healthy individuals without IBD, there's a big difference in the genes which are expressed, and the microbiota actually in the gut as well. And people have shown, certainly in both in IBD with or without liver disease, there's this general picture of a reduction in diversity of bugs and a change towards the more nasty bugs. But there are differences between PSC IBD and people who have ulcerative colitis without IBD in the expression of the actual balance of bugs. And this chicken and egg question always happens. Is this a causation or an association? Is this something that causes a problem, or is it just a consequence of something else which is happening in the liver in relation to bile acid, which is causing an issue? But yeah, apart from Nabil, other people have found the same thing, that there are differences in the microbiome in PSC compared to healthy individuals, both in the reduction in the good guys here. I'm not going to, which is the Bacteroides and Firmicutes I was talking about earlier, and an increase in the Proteobacteria, Streptococcus haemophilus, the Fusobacterium, the pathogenic inflammatory, and also a difference in the bile acid conjugating bacteria as well. And Nabil was able to show this quite clearly. There's a difference between patients with IBD and PSC versus healthy controls as well. So how do you modulate the microbiome? So there are three ways. You can either bung in broad spectrum antibiotics, so you can subtract bacteria, like for example, vancomycin, which I'm going to talk about in a minute. Quite often antibiotics make things worse, particularly in for example, C. diff, which I think people will know about, Clostridium oides diffuse infection. The more antibiotics you put in, the worse it can be. Bacteriophages, of which there are more viruses in the gut than there are bacteria, would be a very neat way to manipulate the bacteria in the gut, but our science is not up to it. We still don't know the first thing about this, really. And then diet, postbiotics, so putting extra bacteria and all these stuff you can buy from Holland and Barrett's. There's nothing really effective there. FMT, fecal microbiota transplantation, which is the kind of crazy thing that I do, is the most effective way of changing a gut microbiome, because it takes a whole microbiome and replaces it with another microbiome. And eventually we will get through FMT, through our science, towards rational live consortia of bacteria, which will be helpful, but we're not there yet. So vancomycin, let me talk about vancomycin. So generally, the treatment of the IBD in relation to whether you have PSC or not is the same as other IBD. It gets nuanced after you've had a liver transplant because the immunosuppressants people are on, but that becomes a very complicated conversation. I think it's probably a little bit beyond what I was going to talk about today, although I'm happy to take questions on it. Vancomycin, there was a small series which suggests that vancomycin is helpful in the IBD side of PSC-IBD. So this small study had 17 patients with children with PSC-IBD. All of them, all 15 who went through to it, their fecal calprotectin, which is the marker of non-invasive marker of inflammation. In children, it's not so easy to do regular endoscopies, but that non-invasive marker normalized from very high to normal in all of them after a period of vancomycin. And this is another small study here showing there was a reduction in this Mayo score, which is the non-invasive score of IBD just based on symptoms. So we did a study, again, led by Nabil, in 15 patients with PSC-IBD and gave them vancomycin for 4 weeks and looked very carefully at what happened at the end to their endoscopic appearance of their colitis, to their clinical scoring of colitis, and then what happened to the metabolites, bile acids, and the microbiome after this 4 weeks treatment, and then 4 weeks later after we'd stopped. And it was quite remarkable, really, in that this is fecal calprotectin, which is the marker, again, that stool test which looks for inflammation in the colon. It got better in everybody in 4 weeks. And after 4 weeks, it started to rise again. So vancomycin clearly has an effect. The partial Mayo score, which is the clinical score of PSC-IBD or any IBD, again, universally got better, but then started to rise after vancomycin stopped. And the endoscopic appearances, how it looked down the endoscope, again, all universally got better at 4 weeks. Obviously, we didn't rescope everybody at 8 weeks, but after 4 weeks treatment, it got better. There wasn't any difference in the liver blood test because, anyway, first of all, 4 weeks is a very short treatment. And secondly, a lot of patients in this study are quite far down the line in terms of the inflammatory process. You wouldn't expect a big turnaround quickly after 4 weeks of antibiotics. But anyway, I'm more interested in the IBD angle. But if you look at the microbiome in these patients who are treated with the vancomycin, it's shot to hell. All the good guys are dead. All the bad guys have gone up. And it's not a healthy looking microbiome, this by any account. So in those patients who get antibiotics, all those nice fiber digesting bacteria have gone down, and there's Enterobacteriaceae, the E. coli, all this stuff, Shigella, goes up. So very paradoxical. But again, I think the important thing is the metabolic pathways, the bile acid pathways, are definitely different here. So there is a complex interplay between the microbiome and bile acids, which we still really don't understand. But certainly after 4 weeks of treatment, this enzyme, which breaks down primary to secondary bile acids in the gut, was greatly reduced. And again, that bounces back afterwards. And there was a correlation between how much of this enzyme, which breaks down bile acids in the colon, there is, and fecal calprotectin. So the more of this you have, the higher the fecal calprotectin. So although IBD is really important in PSC, and I think it drives PSC, the actual mechanisms are different when the liver's involved, and bile acid pathways are really important. We still don't really understand. I think it's quite interesting data. Then this is a study actually which was published just a few months ago in Alimentary Pharmacology and Therapeutics, where this isn't a randomized, this isn't a trial, but this is the Pediatric Consortium database, which consisted of 1,500 patient data from 1,500 kids, mean age of about 12. They were given, there was a look back. This is a retrospective study looking back to see which of these kids had been exposed to more than 3 months of vancomycin and had at least clinical data with regard to their IBD. And you can see that just in this left bit here on the left, these two columns between those patients who had more than 3 months of vancomycin and those who hadn't, there was a big difference in the remission in the IBD. So this prompted an editorial in this journal to say, well, while further well-designed randomized controlled trials of microbiome-treated targeted therapies for PSC with precisely defined endpoints, i.e. liver endpoints, are important, it is prudent to incorporate oral vancomycin when conventional treatment options fail. So there's no harm in going on to this stuff, but got to be we don't have the data yet to kind of just use it as a standard treatment. So, quick word about FMT, if I may. And I'm
Of
going on,
course,
right?
yeah.
It's 10 minutes, 5 minutes, I'm done. So, fecal transplants are when it's really, you take a donor, you screen them as carefully as you possibly can, according to an MHRA guidance, and then you process the stool and you freeze it in your freezer, and it can be used to treat people with inflammatory conditions, mostly Clostridioides difficile infection, which is the horrible condition where you get diarrhea as a result of usually broad spectrum antibiotics. It's usually elderly people. So we went on a journey from 2013 to 2017 to get the first MHRA license to do fecal transplants in the UK, and I've treated more than 1,000 people in that time with Clostridioides difficile. And this is my mate Peter Hawkey, who was kicked out of the university for no good reason, but he started it all. He was an expert in C. diff. So this is our lab, which is again unique in the UK, really, in the public sector, for having a license to do fecal transplants. And for Clostridioides difficile, which is it works amazingly well. This is the systematic review published quite a few years ago now. These here are the case series at the top, and these are the randomized controlled trials. This dotted line represents 90% cure. So you can see it works remarkably well. And it's now NICE approved since 2022. So we're doing quite a lot of it. FMT has been used to treat ulcerative colitis. So this is patients with IBD, but they excluded patients with primary sclerosing cholangitis. The first 4 trials that were done show that compared to placebo, there was a 30% remission rate compared to 10% in the placebo arm. So quite a good signal, really. But they weren't standardized. There's still quite a lot of work to be done. And we did a pilot then in ulcerative colitis, open label. This is our first patient. You don't have to be a gastroenterologist to show that looks quite inflamed. And then after 8 weeks treatment, this looked like this, and we showed. Yeah, I'm not going to make too much of this because it was open label, but we had a 75% response rate, and we've now got some data to show that there are particular strains of bacteria which are important in achieving this effect, and we need more trials. But the Fargo trial, which is open in Birmingham now, and also in Oxford just opening, is the same protocol as the stop colitis study. So we have here it's a randomized control trial. So we have a control group who get an identical placebo, and a treatment group. It's a 1-to-1 randomization. You have a colonoscopy, which times with your surveillance colonoscopy for PSC, which people have annually. And then you either get a colonic delivery at that time of the FMT or a placebo if it's blinded, and then come back every week for 7 weeks for an enema of either the FMT or placebo. And the primary outcome measure actually here is change in alkaline phosphatase, so liver enzyme improvement. Secondary are liver enzyme changes. Other liver enzyme changes, fibrosis, IBD, quality of life indices, and then the stuff that I'm particularly interested in, which is changes in the microbiome and the immune response in the gut as well. So we've recruited 10 so far in 3 months. We've opened Oxford, should be opening in London and Norwich as well. But one of the biggest scandals in the NHS is how long it takes to open trials, like half a lifetime. So I think this is going to be quite interesting. I'm going to stop there. Okay. So
I was going to say we've got a list on our website, on our clinical trials page for Fargo, which will list what sites are open and when they open, who you need to contact.
Yeah. So PSC is definitely associated with the gut microbiome dysbiosis and an inflammatory response. It's strongly linked to IBD, and I think that it's driven by the gut. I mean, I have to think like that because I've been exposed to this for a long time. I've seen how the liver responds when the gut gets better. And liver outcomes are directly related to IBD outcomes. I'm quite convinced of that, really. I think it's very important to address IBD aggressively in PSC, even in the presence of minimal symptoms, really. And this is why it's so important to have that regular surveillance colonoscopy. Not just to look for cancer, because that's the vast majority of people don't get cancer. It's about treating it properly.
Yeah.
Vancomycin's interesting. I wouldn't use it first line in PSC-IBD because the other IBD, proven IBD treatments work very well. We don't have any data in the long term because any antibiotics can cause the growth of multi-resistant germs, right? And vancomycin resistance is very important. And we don't have enough evidence to show that that doesn't happen. And there isn't any real evidence for the effectiveness of vancomycin for liver disease. But for IBD, and I have done this for patients who, basically, we don't have anything else which is working. We need to think where we're going. For a stopgap to fix it, I don't think there's any problem using vancomycin for short periods of time. And then I think the question of how the gut microbiome manipulation will affect liver inflammation, which is what the Fargo trial is addressing, is both appealing and exciting. So that's me. Thank you very much for listening. I hope that's okay.
Thank you very much for that presentation. That I think touched on lots of different aspects that I know we as a charity and the people we interact with are very interested in. The vancomycin for use in IBD is very interesting, and at what stage you should use it. And I think, like you said, the Fargo trial is particularly exciting. If it is, I'm about to say simple. If it's as simple as a fecal transplant, I'm sure it's not simple in the background. But from a patient's point of view, if this is something that happens and there's a significant change, it's a very exciting prospect to happen. We've got lots of questions coming in. We almost certainly won't get through them all, but I will start with one which I think you covered, so it should be a nice, easy, quick one, which is which came first, or which comes first, the IBD or the PSC?
Yeah. So the vast majority of cases, the PSC is sort of picked up first. And the IBD kind of develops later, but that's not always the case. And the problem is we don't have much data on inception cohorts. We don't see people as soon as they present. And what we really need is studies from childhood onwards to follow people to see who gets it. So usually it's the PSC first.
That makes sense. I'm going to combine. There's been a few questions about gut bacteria and how what would be the best way to restore your gut bacteria, say after a colonoscopy or after a course of antibiotics, or if that's even possible, if changing your diet or eating a certain thing can actually influence your gut bacteria.
Well, it's a long game, this one, in terms of diet. And unfortunately, there are some people who can't tolerate the type of diet that will be best for the gut because of the actual inflammation that's there at the time, right?
Yeah.
But if you don't have strictures in your gut, there's no narrowings, it's not a sort of Crohn's type thing, then taking a decent amount of the kind of fiber which we can't digest, but our bugs thrive on, such as pulses and broccoli and this sort of stuff, is very beneficial. It's pretty clear that large amounts of fat and red meat and processed foods are definitely detrimental to the gut microbiome, as is white sugar. So that from a diet point of view, in terms of probiotics, the stuff you buy from Holland and Barrett, I don't think they don't really touch the sides really in this place. But on the other hand, I don't think they're harmful either. So it's okay to do that.
And so if you find that it works for you personally, fill your boots.
I think that's fine because there's only, as somebody has written here, I've read one of these, that there's only like 10 to 12 species of lactobacillus and bifido generally in these things, and that's really a drop in the ocean in terms of what's in the colon. Yeah.
Makes sense. One question we got in beforehand. I know nothing about this, which is terrible of me. I should know more. But someone was asking if hormone replacement therapy to help with perimenopausal symptoms, if there's an issue there, because their understanding is that estrogen, so the estrogen they get through a patch, and the guts don't tend to get along, is how they phrased it. Does this pose a particular challenge, and is there anything that they can do to sort of help that along?
Well, there's epidemiological evidence that there is a sort of weak association between estrogen replacement and triggering IBD, but it doesn't happen in everybody, right? So you've got to be pragmatic about these things, because there's no one clear mechanism, and so if you've got really bad menopausal symptoms, then you'll find out. If you take it, it'll cause a problem. Stop it, it'll get better. So it's caution, I think, is the word. Yeah.
That makes sense. I'm going to combine another couple of questions we've got. One we had beforehand and one that someone's asked. But in your view, because this can only ever be in your view, someone's asking if they had a transplant and then they underwent chemotherapy. They had lots of antibiotics and lots of medications that acted on the gut biome. Could that have contributed to their recurrence of PSC? And we've also had a question about, is it possible for things like the COVID vaccine to affect or trigger PSC?
On the second one, there's no evidence really on that one at all. But in terms of I think it's quite clear that after a liver transplant, if your colitis is active, that's not good for the liver that's there. It's not going to be dramatically terrible, but it's really important to get on top of that IBD. And the other thing is, never have a pouch if you can help it after it.
Okay.
Yeah. But,
so that actually leads into another question I had here, which is, will the PSC disease calm down after the colon is removed? But that suggests that isn't the case.
No, I think we don't know exactly. Okay.
Of course. Yeah.
So what I'm kind of telling you is what my experience is, and also if you had my colleague Dr. Trivedi sitting next to me, Prof. Trivedi, he would say exactly the same thing, that we've both seen that if you can get the IBD nicely under control, the liver enzymes get better. Okay, but it kind of depends on probably how far down the line the liver damage has got, and how much of the problem is related to recurrent cholangitis as opposed to being driven by the IBD. But certainly, having inflammation in the colon doesn't do the liver any favors at all. Does that answer the question?
I think so. Yeah, I understand that a lot of these questions are kind of pushing against the edge of our knowledge of PSC anyway. So
it's also very individual dependent as well. Because this is not a one size fits all type of disease. There's a pathway.
I'll do this one quick question and then one final one. There's a question, I think it's referring to the Fargo trial, which is, can children under 18 access the Fecal and microbiota? I don't think the Fargo trial allows under 18s.
No, it doesn't, I'm afraid. I have an IBD PSC IBD inception trial, which I'm just waiting for the result of funding. And that'll be the only one, the first one to have children in it, actually. So,
okay.
Yeah.
So there might be something coming in the pipeline, but right now, unfortunately, it's not.
There might well be, yeah.
Okay, and the last one was just one that piqued my interest, which was about the sampling of the microbiome. Is that done in the large colon or the small intestines? And is there potentially a difference between the two? They were saying that they'd seen something that suggested there is.
Yeah, so the small intestine is supposed to be sterile, and it isn't sterile, but it really is sterile in relation to the colon. The colon has got so much more. It's got so much more bacteria than the contribution from the small intestine is pretty minuscule, really. So we're looking at the gut microbiome with fecal samples, and that reflects the small intestine and the colon, but the vast majority there is related to the colon. But more importantly, it's important to look at the bacteria which sit on the gut wall, the mucosal bacteria, and we're taking samples from that as well throughout this trial.
Excellent. I'm going to call it there because we're rapidly running out of time. I have to say this is the most questions I think I've ever seen come in for a session. So maybe some point next year you might be able to join us again for another one of these sessions. Maybe when we're a little bit further along in the Fargo trial. I think that would be particularly interesting.
Yeah, I'd love to do that. And yeah, so thank you for asking me to come along. I enjoyed that. And hope I didn't hang on too long.
No, it was fascinating. At least from my point of view, I can't speak for anyone else watching, but I found that fascinating. It was lots of information I didn't know about my own gut, and I found that brilliant. You've got lots of claps coming up from the chat at the moment. So thank you again for joining us. For those of you who are listening, we do these every month. We've got one next month. Off the top of my head, I can't remember what next month's one is. Oh my gosh, that is terrible of me. I should have checked before I came online. But there will be a little survey that goes out after all of these events, so you can let us know what you think of it. The next month's is on. It's not actually next month. It's just into December. Monday, the 2nd of December. It's with Dr. Jessica Dyson, who is a consultant hepatologist with the Freeman Hospital in Newcastle, and she's given us a history of PSC talk, which I think will be absolutely fascinating. Once again, thank you, Professor Iqbal, for joining us today. It's been absolutely wonderful having you here. And I won't be seeing you in clinic in the future because you're now wholly dedicated to research, which is a definite win for research. So thank you very much.
Thanks, Mark. Really enjoyed that. Thank you.
Have a lovely day. Bye-bye.